Very good day to everyone. My name is Neha Priya. I'm working as assistant professor in IIMT College of Pharmacy. Today, I will be introducing to a new topic that is Introduction to Drug Discovery and Development. So before moving further, let us discuss about the contents of today's lecture. So today's lecture will be comprised of introduction along with the history of drug discovery and development, the process involved in the drug discovery and development, drug discovery and development timeline, and the last will be drug discovery process. Like what are the important factors that are involved in discovery process? So let us first understand what is drug discovery. So basically what happens is whenever there is a new certain disease, for an example, like as everyone know about COVID-19. So when COVID-19 came, there was a need of a drug to cure that disease and to cure and to like have the drug we were like comprised of a certain techniques, we were having certain techniques, we were having certain criteria to develop a new drug to like have this new drug and use it as in the treatment of COVID-19. So they basically drug discovery and development is a very important process, because if will be no discovery or no development in the drugs, then we will not be able to cure the disease which are arising now. As we all know now, the pollution is more now, the lifestyle is more worse than earlier, so we need to have certain developments in the drugs to cure ourselves, to maintain our healthy lifestyles. So basically what happened is, in past drug were like in past there were no drugs, so the main motive at that time was to like have a new drug, to like discover a new drug, to identify a new drug. But later on, what happened was we were all in a severe position when we were having that drug, but we were not able to cure a certain kind of disease. Like we were not able to cure a severe disease. So what we needed was we need to modify that drug to further use to further like use that drug in a better way, in a better conditions, in a better treatment, so that we can be cured easily with more severe disease than before. So the history of drug discovery, basically what happened was in pre-1919, only thing that happened was we were just identifying the drugs. We were just knowing that this drug can be used in the treatment of a certain disease. So the only herbal drugs were identified, and basically we were more towards the fact of serendipity. Basically, what does serendipity mean? It means nasib. Basically, if we if I am having that drug and we were having like using that to cure a certain disease, if we were lucky, then the disease was cured. If we were not, then we were moving to the next drug, like identifying then the next drug and then curing the next disease. Then later what happened was vitamins and vaccines were developed in 1920s and 30s. Then in 1940s, there was basically antibiotics era, we can call it, as in in World War II, what happened was everyone was have suffering from certain disease because of food, because of lifestyle, because of water, and because of like the conditions, everyone was suffering from certain disease. Then there was a major like development in that era, because at that era everyone was trying to like get a cure of that disease, and in that era we can call it as an antibiotic era. So the major class of drug that was identified in that era was antibiotic. Later on, 1950s, new technologies and discovery of DNA was done. In 1960s, breakthrough in etiology. Basically, what does etiology mean? It means that our life is like there is a condition like epidemiology, air, environment, host, and the guest. These all are responsible in creating a disease. So basically, we were more towards discovering the etiology of a disease, etiology of the conditions. Then in 1970s, there was rise in the biotechnology, and the use of IT was introduced in our systems. In our like discoveries, we were more prone to like use the biotechnology to discover new drugs. We were more used in the like identifying the new drug via our technical technology. For example, HPLC, IR, we were using that to identify the certain structure of a drug, if that drug can be used in a certain disease or not. We were using biotechnology for that. In 1980s, what we did was we then move toward the commercialization of drug discovery. We now, at the time, we were like, we have identified a major class of drugs. We have identified like 1,000 tons of drugs, but are we able to use that drug rightly, or are we able to use that drug in their full potential? So in that era, we like felt like there is a need of what drug discovery and development. There is a need of discovering a drug or developing a drug to like use that in their full potential. And then further in 1990s, what we are doing is we are more towards AI, robotics, and everything, so that we can develop a new drug that will be used in more severe disease that can be, helpful in curing more disease and majorly giving us a better result in the treatment of every disease. So this was the history of drug discovery. Moving towards what is drug discovery, so I'll quote the quotations, but that is a process that starts with the identification of disease and therapeutic target of interest and include mathematical methodology, assay development, lead identification, and characterization in vitro, formulation in animal pharmacological studies, pharmacokinetic and safety studies in animals, and clinical studies in humans. Basically, what does drug discovery means is first we are going to identify a disease. When I know the disease, then I'll identify the target that is responsible in like creating that disease, and then I'll discover a lead that will go and target and go on the target and make sure that that target is cured. Like that target in that lead is working on that target. If that lead is working on the target by which the disease is called, then ultimately what we'll be having is will be getting a better cure for that disease. In this phase, there is a clinical trials, preclinical trials, and every trials is going on this in process. So basically, what we do is first we do the research part. First, I'll like get to know about my target, about my lead, like which lead can be potentially have certain factors on that target. Then what I'll do is I'll work on this. I'll create a program. I'll synthesize a drug or create a drug or develop a drug. Then further, there will be preclinical trials and then clinical trials. And after this, the drug discovery can be done. So elaborate that, I'll show you a graph. For an example, there are like 1,000 compounds for a certain disease, out of which only 250 compounds will be able to cross the preclinical trials, and out of which only 5 compounds will be able to overcome the clinical trials, and only one drug will be approved. So it is a very lengthy process. For an example, I am like having tons of drug in the initial research. Then further, I am like uh, continuously, making my way through the one drug that will be going to be like the major, which is going to give the major impact on a certain disease. So it is a very lengthy process overall. So the drug discovery process consists of 6 parameters. The first will be target selection. First, I select my target. If a person is suffering from a disease, then first thing what I'll do is I select the target which is responsible for creating the disease. Then further what I'll do is I'll select my lead, which can act on that target and able to cure the disease. Followed with medicinal chemistry. In this medicinal chemistry, what we do is we do our in silico screening, we do our library development, we do our SAR studies to make sure that that drug will be able to cure that certain disease. After that, we'll go with in vitro studies of that drug, then preclinical trials and clinical trials. For that, I'll be elaborating this points to make sure that everything will be well understood. So in drug discovery process, the first thing like drug discovery method consists of 4 methods. First one will be the random screening along with molecular designing, drug metabolites, and serendipity. Earlier, people used to have this serendipity the more because they believed in the seed. They were not having that technology to work on, molecular designing and everything. So they believed in serendipity. Later on, we moved to random screening. We just used a random target. We will just use the target and this had like a big number of compounds to work on that target. And if that compound is giving a certain result, then we were okay with that. Moving further, there was drug metabolites and molecular designing. Molecular designing is a very good, initiative in the drug discovery, as because of this molecular designing, we get to know or understand the drug in a more better form than in the other states. So basically, in earlier things was like going in a traditional manner. Then later on, current target-based discoveries happened, and after that, system biological approach was there. So basically, in traditional manner, what happened was first we first we'll do the bioassay, we'll get the biological hit of, then the lead optimization will collect our lead, will identify our lead, then there will be clinical trials, preclinical trials, and there will be success rate. So in the success rate in this manner was more because first we used to have the target, and we were going with the leads that will for sure work on that target, and then we were doing our preclinical, then clinical, in vitro, and every study. So the success rate in this manner was more rather than in first in this current target based discoveries, because there was a large number of drugs used in this manner, and system based is also a successful rate because there is a certain pattern which we used to work with that. Basically, first we need to understand about the biological assay, because if we know about our target, then only we'll be able to understand our lead, or what kind of lead will be responsible, or what kind of lead will be good in our treatment. So understanding our target is a very important topic. So the first topic in rediscovery itself is target selection. What does target selection means is, for an example, if a person is suffering from a disease, then certain target is responsible in creating the disease, in making this disease happen. If we get to understand this target, like how it is creating that disease, if we get to understand the MOA of that target, mechanism of action of that target, then for sure we can block, we can, make this target not work in that manner by our lead. We can, like, from thousand of leads, we can choose one or two leads, or like if we are going with like 10,000 of leads, then we can choose 1,000 leads in that manner which can work on that target. So the area of research will be also confined. And the second thing what will happen will be, we will be getting better leads because we have already understand about our targets. We have already selected our target, that this target will be responsible for creating that disease. The first thing will with target selection will, we will start like identify our select our target, then we will identify our target. To identify molecular targets that are involved in disease procession, we will identify our molecular target, identify our target, and what we'll do is later on we will target validation. Let us, if we work on that target, then the disease will be for sure cured. We'll do that. Moving on, like there are plenty of targets that are in our body. So we need to identify the mode of action of a certain target that is responsible in creating that disease. If I'm not able to get that, identify my target, then I'll not be able to get a perfect lead for my target. For an example, if I am not having the full knowledge of the target, I'll be selecting a lead that will be also on a partial knowledge. This will make my discovery a more lengthier discovery because I'm not sure that that target will interact with my lead or not. If my target is not interacting with my lead, then for sure there will be very less, we can say there can be a major failure in this process. So the first thing that we need to understand is target selection, and target selection needs to be done in a very gentle and in a very collective manner, so that we'll be able to work finely with our other discovery stages. So the drug target selection can be done by four, or are distributed into four categories. First one is cellular and genetic target, along with genomics, proteinomics, and bioinformatics. These are the four factors, four types that are involved in target selection. First, we'll go with the cellular and genetic targets. What does cellular and genetic target means? Basically, it is the identification of a it is the identification of a function of a potential therapeutic drug target, and what are the roles of these drug target in the disease. It can be Basically, what we do in this is we select a drug that will work either on a cellular or a genetic chemicals known in our body, and these cellular and genetic chemicals are, seen as a potential target that are working in a system. Basically, this can be, mapping genes or sequencing of the DNA, and these all come into this cellular and this target. Further, we'll be moving towards genomics. Genomics aims to understand the structure of genome, including the mapping genes and sequencing the DNA. These all are coming to into the genomics, and there, if I am knowing about my body parts, then I can, if I'm knowing about the cell, then I can make a lead that can work on that cell. If I'm not understanding the concept of my target, then I can't move forward with the other stages of drug discovery. Then the third one is a proteinomics. Basically, it helps in identifying the protein that is responsible in drug development. So proteins are very important because basically whatever we are doing in a docking study is basically we are creating a target as a protein, and then we are making a drug and we are interacting. We are seeing the interaction of our lead drug with our protein and making sure if that is giving us the best results or not. So, in fact, proteinomics is a very important factor in a target selection. Last one is bioinformatics. Basically, what does bioinformatics is? A molecular biology that involves extensively analysis of a biological data using computers for the purpose of enhancing biological research. It is a very important factor. In this, what we'll do is we need the information of our of our targets, right? In this, we'll be able to get that information. If I'm getting this information, then my target selection is done. First thing in drug discovery is target selection. First, I'll select my target, identify my target, and validate my target. If this process has been done nicely, then only I can move to the next stage of my drug discovery, that is lead discovery. So basically, what does it mean? Lead discovery mean after identifying my target, after looking towards my target, after knowing my target, I'll move toward the lead discovery. That is, I'll be making sure to understand the lead compound that can be able to cure my disease. Basically, it can be small molecule or it can be a large molecule. Depending on my target, I have to select my lead. And that lead needs to be, selected in a more focused manner, because that lead needs to understand that lead needs to be, able to confirm that it is working on my target. Later on, we can have the other parameters. For an example, viability. First, I'll make sure that that lead is responsible in curing that disease. That lead isn't responsible, like hitting that target. If I know that, then I can work on the other parameters about the viability of a compound and other things. But the first thing I need to understand is that that lead needs to work on that target. So first thing I'll do is I'll select my lead and I'll synthesize and isolate my lead, because we can't use a drug in a crude manner. We need to find out the main ingredient or the main active that is working on a target. For an example, if I'm going with quercetin, then I need to know, for an example, I'm going with flavonoid. Then I need to know that in flavonoid, quercetin is responsible for hitting in that target, and I need to isolate that quercetin. If a certain green group needed to be sampled in quercetin, then I'll do is I'll synthesize that. So first thing is target selection is done. Then further I'll make sure my lead is selected. After selecting my lead, I'll do is I'll either isolate or either synthesize my lead. What will happen is after this, basically I'm like separation of mixture. I'm separating my lead. I'm if my lead is like whole flavonoid thing is present, I'm not selecting just my flavonol part, that is quercetin. I'm selecting my lead. And after synthesis and isolation of lead, the next thing we'll do is combinatorial chemistry. But moving further, I need to you need to understand about serendipity. Basically, in earlier centuries, people used to work on serendipity. What does it mean? It is basically, nasib. If I'm like having a lead, I'm making sure if possible, that will be able to cure the disease. If not, then that will not be able to cure that disease. So basically, serendipity low is this, but the chances and success rate in the serendipity was very low as compared to other thing. But one of the two of the most important drugs which we are using in today's life, where proic acid and everything was selected by this method. So indeed, this method was successful hit at that time. For that time, it was a successful hit. But now, as we are working on AI, as we are working on molecular docking, molecular modeling, and everything, then we need to not depend on serendipity at this level. Then basically, what is rationale drug design? Basically, in rationale drug design is there are plenty of drugs present in our world, like plenty of drugs present in our world, but out of which we need to synthesize the exact amount of drug which will be responsible in, like for an example, I have given you the example of quercetin. I need to select out of flavonoid, I need to select that quercetin, that this part of flavonoid is responsible in creating, in curing my disease. So this part need to be selected by that. This is what needs to be done in drug discovery. Then the next point is combinatorial chemistry. Basically, in this is basically we use computer stimulation of large number of different but structurally related molecules. For an example, that drug is working. Quercetin is having effect on anti-cancer, but the viability of quercetin is low. So what I need to do is I can synthesize quercetin in a manner that its viability get increased. Like there need to be increase in viability of quercetin. If I work in that manner, then the viability of quercetin will be increased to a certain point. So basically what we do in this is we search for new leads, or we work on the new leads, identify their selectivity, target affinity, we calculate their ADME properties. What does ADME means? I'll quote it one more time, that needs to go with ADMET. Basically, it stands for absorption, distribution, metabolism, excretion, and toxicity, which we can get through with Swiss ADME, MolSoft, and PKCSM. By this software, you can analyze the ADMET properties of any compound, and the drug needs to be reduced, toxicity needs to be there, and eliminate side effects. So these are the points that needs to be taken care while we are selecting a lead, or we are validating, or we are making sure that lead is working in a proper manner or not. Next point is assay development. So in assay development, basically is we after continuing with our lead is synthesized, our lead is done, then we need to measure the activity that can be done by the drug. These come under the assay development, and the most important part is high throughput screening. Basically what is done in this is, in this we can select a large number of drug, and approx 50 to 100 a large number of protein, and we can run this or screen this drugs, large number of drugs at a certain time. So this screening is very important, as we can at that time we can search over a large number of drugs, we can get the result, and we can screen that drug, large number of drugs in a particular time. So this is a very important factor also, because for if I'm going with MolSoft, because yes, I'm just selecting one drug and just screening that. But in high throughput screening, you can select over 1 million of compounds against 50 different proteins and screen them at a time. So it is basically a very very important part of lead discovery. So our lead discovery is done, our target selection is done, our lead discovery is done. Next one is our medicinal chemistry. What we do in this is basically we synthesize a drug by synthetic organic chemistry. We understand our this SAR study. SAR, I think you have heard about structure activity relationship. Earlier we used to have structure activity relationship, but there were certain limitations with structure activity relationship. For an example, I was not able to get the optimum distance between the bonds, and this distance between the bonds also is a very important factor in very important factor in working with the target, because this can also contribute a very vast changes in our response. So it is a very important factor. So working with this is a very, fine thing. I can go with SAR, but the more advanced version, like QSAR is the term, that is qualitative structure activity relationship, that will make us understand the distance, that can make us understand the van der Waals force and everything within the lead discovery. So these also needs to be done, or these also needs to be understood by us in the whole process. What we'll do is we'll high screening throughput. Will, based on organic chemistry, we'll do the QSR studies based on collection of stored chemicals and all along database. We can use that in select the process which can be used in the synthesis in the synthesis of our novel drug. We can do that. We can identify the pharmacophore. So what does pharmacophore mean? Pharmacophore means is basically the most active part in a molecule that is responsible in like interaction with the target, or that is responsible in giving us the optimum response. That is our pharmacophore. Then continuing this with, so basically this medicinal chemistry, as earlier I told you, is comprised of library development, SAR studies. We have already discussed about the SAR and QSAR studies, along with in silico screening and then chemical synthesis. So in silico screening is basically I get to understand about my drug by the ADMET of the drug, by certain factors, that is our PKCSM, EDME, and everything. We get to understand our drug. Then the next part is chemical synthesis. So chemical synthesis is basically in this part, this medicinal chemistry, we are going to understand about the chemistry of our drug. We're going to understand what is the main factor that is responsible in creating that drug. What is the main pharmacophore that needs to be, protected, and what are the changes that needs to be done to get the optimum result. So these are the all things that needs to be done in the medicinal chemistry sector. Moving on with our in vitro studies. In vitro studies, we just, get to know about the drug affinity and selectivity, cell disease models, MOA, and lead candidate refinement that needs to be done in vitro studies. These needs to be understand. The ranges and the parameters. These need to be taken care in our in vitro studies. Then the next one is our clinical trial. As we already know, before any clinical trials, no drug can be introduced into the market. No drugs can be getting any approved way. It needs to be done in a better manner. We need to have, preclinical trials and then clinical trials. Then this preclinical trials is composed of animal models of disease state, behavioral studies, functioning imaging, and ex vivo studies. In this, what happen is first we'll do is pharmacological testing, then preliminary toxicology testing, pharmacokinetic studies, that is ADME studies, and then chemical and pharmaceutical assessment of the drug that is done in the preclinical trial. Then the next, the thing is, what we do in this is we get the toxicity, drug interaction, if that drug is interacting or not, the toxicity of the drug, and the metabolic profile of this drug. We get to understand about these in the preclinical trials. After understanding, the preclinical trials, we can move forward to the clinical trials. But in preclinical trials, there are certain factors that needs to be taken care of. In this, the first one is animal model of disease state. What happened in this is we test the condition, what is the toxicity level of this drug. For example, if an animal is suffering from this drug and I'm giving that drug, then what is the optimum of toxicity that can be caused to an animal? Is an example of a certain point that needs to be tested in this. Then the second one is behavioral studies. Like if I'm giving a drug, then are there any side effects or the behavioral symptoms that is seen in the animal? If that animal is not is repelling to the disease, if it's showing something different, then these all needs to be taken care in the behavioral studies. Then the next one is functioning image. We get the functioning image of everything by the help of MRI and CT scan. We get to understand the drug behavior by this. Then the next one is ex vivo studies. In ex vivo studies, the external parameters, the artificial environment in silico, we can say that artificial environment is given in the outer manner, and the drug is accessed by the help of computer aided drug development. Then the last one is the clinical trials. Clinical trials comprised of 4 phases, that is phase 1, phase 2, phase 3, and phase 4. Basically, in phase 1, what is done is we just get to know about the safety and efficacy of a new drug. It may require around up to 7 years to like access this phase only. Then earlier after that, we evaluate about the human toxicity, like what toxicity is given to what toxicity is happening in the human, moving on with tolerated doses range, and then pharmacology and pharmacodynamics studies. Then the next in phase 2 is what happened is, in this phase we are more concerned towards the safety, interaction of drugs, and efficacy of drug. In earlier manner, we were just using what does that drug is doing, In the second phase, we were working on efficacy, safety, and everything. In third phase, what we are giving is we are giving a double blind technique. We are using in this phase is what we are using a double blind technique. It is given to 250 or 1,000 patients, and it is the most consuming and time taking phase. After there, the last phase will be the market surveillance phase. We get to understand the adverse reaction of a drug. So basically, these all are the parameters that need to be taken care in the drug development. I'll quote with one example. For an example, I have to select, if suppose I have to select developer this developer medicine for anti-cancer. So the first thing I'll do is I'll first select my target, like which cancer target I'm selecting. After selecting that target, I'll choose a lead that will work on that target. After selecting the lead, what I'll do is I'll make sure if I need to, develop, I'll read about their SAR studies, their QSR studies, and after knowing that, I'll develop a method to synthesize my lead in a manner that will work in a more efficient manner towards my target. After the synthesis, what I'll do is I'll do the testing work, that is in vitro, preclinical, and clinical. And if my drug is standing out in this trials, then my drug will be FDA approved and will be given into the market. So this basically drug development and discovery is a very important phase, as we are already knowing majorly we are having many herbs and many herbal treatments present with us, but we are not able to use that herb in their full potential manner. So we need to understand their potential. We need to develop that herbs in so that it will take less time, because discovering a new drug is a time taking process, but developing can take lesser time than discovering, because in discovering you are first making sure you're in a search of a new drug, but in development you are already knowing that drug. You just need to work on that drug to get that efficient result. So drug discovery and development is a very important process, and we need to work in that phase. Well, thank you for patiently listening to me and giving me your time. Thank you very much.